All right, so I handed back the clotting-related homework to you guys, and I think you guys, for the most part, did a really nice job, of that. And in some cases, I was pretty lenient with the grading, especially with talking about how when you guys talking about the clotting cascade, how some people were mentioning how, say, prothrombin activates thrombin there, right? But really, prothrombin and thrombin are two different versions of the same protein. Does that make sense to folks now after how I explained it on Monday? So prothrombin's kind of the inactive zymogen version, and then you cleave off or remove a piece of it, and now it becomes active. Is that clear for folks now? That's making sense? Okay, I wanted to make sure people were understanding that. I know it's a challenging concept to kind of understand if you didn't have that background. So I want to make sure that people were clear on that now. And then for the in-class quiz, so hopefully it motivates some folks who didn't do as well as they had hoped to go and review that material. So if you haven't reviewed that material, you're really falling quite far behind if you haven't taken a look at that. And so be sure to take a look at that information because we're going to be building on a lot of concepts that are really important, but there's a lot of moving parts. All right? Okay. So we left off the other day with talking about the cardiac conduction system. And remember, we've got SA and AV node. These are areas where we have electrical impulses emanating from them spontaneously in a rhythmic fashion, right? So here, the SA, we have a piece of electrical tissue, not neuron, it's cardiovascular tissue still, all right, where we generate the sinus rhythm, we say. The frequency of those electrical impulses is about 60 to 100 times per minute. So that gives us a certain firing rate, right? And so remember, the cardiac muscle, when it receives the stimulus, contracts. So here, remember, just like when we talked about skeletal muscle, we needed a stimulus to cause the skeletal muscle to contract. Same thing with the cardiac muscle. We have a bioelectrical stimulus, and it causes the muscle to contract. And remember, the cardiac cycle is the physical movement of blood through the heart, where we have atrial systole, and then we're going to get atrial diastole, where it relaxes, and then we get ventricular systole, and then we get ventricular diastole, and then it goes back and forth. The physical movement of that cardiac muscle tissue, though, comes about because of the bioelectrical signaling to it. So the bioelectrical signal, right, the cardiac conduction system that you can record via the ECG happens before the actual physical contraction. Just like, when we talk about skeletal muscle, the actual contraction of the skeletal muscle happens after the motor neuron fires, right? Okay? Same thing. Cardiac conduction system happens first, and then we get the systole and diastole. Everybody following me on that? Okay, that's going to be important because in the lab and also here in lecture, I want you guys to be able to connect what's physically happening with the cardiac cycle to what's happening in the cardiac conduction system, how they're related to each other. All right? Okay, so here we have some tissues that spontaneously are generating those bioelectrical impulses, and we have the SA node and the AV node, and remember it starts off, that SA node is going to be in the upper part of the right atrium, and it spreads towards the AV node, and then also is spreading towards the left atrium, right? And so you got this spread that goes on, and just like what you guys remember with the nervous system, it's an action potential where you get depolarization, then you get repolarization, right? Depolarization is the signal that is going to be signaling that cardiac tissue to contract. And so here what we're going to, this signal with depolarization that spreads is signaling the atria to contract. So maybe not totally evident in this picture in the lower left-hand corner, but the little green arrows are pointing to the slight contraction of the atria there. Okay, so they're contracting because of the bioelectrical signal that they're receiving. And then there's a slight delay at the AV node. Then the AV node passes that electrical signal on into this bundle called the bundle of His. And then there's branches that go down what we call the interventricular septum. Septums are just structures that separate other structures. And the reason we call this the interventricular is because it separates the two ventricles. Okay? So it goes down the interventricular septum, and then it's going to start spreading. So you can kind of make out that there's some fibers here that are called Purkinje fibers that are innervating, or not innervating, but they're throughout this cardiac muscle tissue. Right? So the bioelectrical signal that's coming down here is going to stimulate the ventricles to contract. Okay? Right? And so that's the series that goes on for every heartbeat. All right? Now, if we get some area other than the AV or SA node to spontaneously fire, we call that an ectopic focus. Okay? So spontaneous firing at some place other than the SA and AV node, right? So we could have, say, this area, for whatever reason, starts having, some sort of depolarization event. It causes that area to contract, right? What's the problem with that? We have an ectopic focus. Throws off the rhythm, right? And so remember here, the heart's a pump. And really, it's a one-way pump, right? So do you think that pumping action is going to be more or less efficient? Less efficient, right? And then we're going to have issues, right? So here we can have an ectopic focus that can that can manifest itself as, say, arrhythmia, all right? Could be. We're just saying, if this happens, it can lead to some other things like that are diagnostic. Yeah. There's other ways you can get to that, though. Yeah. All right. So cardiac conduction. So like I said, the heart itself, the actual beating of the heart, doesn't need actual nervous stimulation. It has its own pacemakers, right? The SA node is the internal pacemaker. It spontaneously is depolarizing to cause, a beating of the heart. But we do have sympathetic and parasympathetic innervation of the heart. These neurons, these innervations, aren't causing the heart to contract, but they can control the rate at which it contracts. And then with respect to the sympathetic, it can also affect the contraction strength. So when we affect the rate of heart contraction, the heart rate, we say that's chronotropic. So when we talk about chronotropic agents, we're talking about agents that affect the heart rate. Right? So think back to sympathetic and parasympathetic. Right? So which one is going to be fight or flight? Sympathetic. So not surprisingly, fight or flight is going to raise heart rate. So we say that's a positive chronotropic effect. Right? So epinephrine, norepinephrine. So it's a positive chronotropic agent. Right? Okay? Now, what about parasympathetic? So rather than fight or flight, what's that involved with? Rest and digest, right? So basically, being, less excited, right? So rest and digest. And here, if we're resting and digesting, then we're expecting the heart rate to go down, right? And so we say, if we said that the sympathetic, or let's say epinephrine, norepinephrine, are positive chronotropic agents, what's released from the parasympathetic? Anybody remember what neurotransmitter? I think you guys got it. ACH. Yeah, acetylcholine. Yeah, so remember all of the neurotransmitters of the autonomic nervous system are all acetylcholine except for the postganglionic of the sympathetic, which is epinephrine, norepinephrine. So you have a good chance if you say acetylcholine or ACH there, right? So ACH with respect to a chronotropic agent, right? If epinephrine, norepinephrine was a positive chronotropic agent, what would we say about acetylcholine? A negative chronotropic agent, right? Because it decreases heart rate. Now, if we have sustained elevated heart rate, we call that tachycardia, right? So the term tachy refers to rapid, and they slow down heart rate that's sustained, right? We call that bradycardia. So brady means slowed down. So a few terms there. Okay. So major parasympathetic nerve, right? Cranial nerve number 10, the vagus nerve. So a vagal maneuver, for those that are in the healthcare field, what you're trying to do is stimulate the vagus nerve to have a negative chronotropic effect. Slow down the heart rate. Because we'll see the heart rate is one component that affects the blood pressure. Okay? All right. So vagus nerve terminates at SA and AV nodes, meaning that it's affecting the rate at which we're stimulating the heart to contract, the rate. Now, let's go back up to the sympathetic. Terminates not only at SA and AV nodes, but also the myocardium. So here, the chronotropic effect is happening because it's affecting some of those nodes, right? And then it also has effects on the contraction strength. Contraction strength, right? So agents that affect contraction strength, there's a term for that too. It's called inotropic, I-N-O-tropic. Inotropic. So inotropic agents are going to affect contraction strength. Chronotropic agents are affecting contraction frequency, so heart rate. All right? So this is why sympathetic can not only affect that heart rate, but also affect contraction strength. Everybody following me on that? Okay? And so here, just like when we talked about the autonomic nervous system, a lot of these organs are dually innervated by both branches of the autonomic nervous system, sympathetic and the parasympathetic, right? And so the question is, which one becomes dominant, right? Okay, which one becomes dominant? And generally what we do is we kind of think about, all right, during the normal resting state, right? Which one has the dominance over this? Well, it turns out that the resting heart rate's lower than the pacemaker heart rate that was 60 to 100 beats per minute, right? So remember the sinus rhythm was 60 to 100 beats per minute? How many folks have 100 beats per minute just resting? Not many, right? And so it turns out that there's some tonic or some stimulus coming from the vagus nerve, the parasympathetic, to constantly keep that heart rate slightly depressed. So when I say tonic, do people understand what I'm saying? Well, with tonic firing just means that there's a steady rate of firing of the vagus nerve, right? So even at resting conditions, right, where we're not necessarily overly stimulating, right? So there's just, and it's very similar to when we talked about the blood vessels, where there was tonic firing to keep some sort of vascular tone. Exactly. So that's exactly the same. So there's some tonic firing that goes on by the parasympathetic to keep that heart rate low. That means that you can raise the heart rate just by simply doing what with the parasympathetic, with the vagus nerve? Shutting it off, right? Because it's constantly kind of putting the brakes on the heart rate. You take the brakes off and heart rate goes up. So you can increase heart rate without stimulating what branch of the autonomic nervous system? Sympathetic, right? Everybody follow me on that? Okay. So we say there's parasympathetic tone here with respect to the heart. Everybody follow me on that? Okay. Okay, so this is what's happening with these pacemaker, these nodes, especially an SA node, right? So remember with neurons, we had a resting membrane potential. That was negative 70 millivolts. Only time we moved from that resting membrane potential was when we had some sort of ligand-gated channel, ion channel, open or close, right? If it allowed positive ions in, it meant that we depolarized. If it meant that we opened up a channel to let positive ions out of the cell, we hyperpolarized, right? Or we could hyperpolarize by letting negative ions in, right? We had to have some sort of stimulus. Turns out there's channels that get stimulated to open right after this repolarization. So as soon as it gets repolarized, there's channels that open, and they are sodium channels, right? And so remember, sodium is higher where? Outside of the cell or inside of the cell? Outside of the cell. And so here, if we open those channels, what happens to that membrane potential? So it depolarizes, right? So that's what ends up happening, is that we get the slow inflow of sodium, so that membrane potential constantly rises until it hits threshold, right? Threshold is a voltage at which ion channels are going to open to cause an action potential. And so rather than just having sodium ions flowing in for depolarization, we actually get a huge inflow of calcium for the depolarization event in the heart, the myocardium. Okay? All right, and these, I should say, these specialized cells that spontaneously are generating that, nodal rhythm. Okay? And so this is what ends up happening. We have slow sodium ion channels open that are constantly allowing that membrane potential to depolarize till they hit that threshold. And then we have some voltage-gated ion channels that open that cause basically what you guys are familiar with an action potential, where we have depolarization and repolarization. And then once we get to the repolarization, and those sodium ion channels open again. So technically, we don't have a resting membrane potential with these cells that we find in the SA and AV nodes, right? Is it ever resting? No. It's constantly moving, right? And believe it or not, back in the day when they were looking at this, they said, oh boy, this is really funny. This membrane potential constantly is moving and depolarizing. It's very different than, say, what we look at when we see motor neurons. So they called these sodium ion channels funny channels, right? And this was the funny current. Yeah, so if you look up, if you want to figure out what channels these are, this is the funny channels, right? Funny sodium ion channels, okay? All right, because they cause this funny depolarization event. Now notice here, compared to what we talked about in the nervous system, calcium is really important for the action potential here, right? So if we start disrupting calcium levels, right? What's happening to this? We're having problems with depolarization, for instance, right? Now, hopefully, if you take a look at this, right, each one of these is signaling the heart to contract, right? So here, if we change the slope of this, right, so if we made the slope of this membrane potential that's slowly rising towards the threshold steeper, right? What's that going to do to the frequency? If we have it's going to be steeper so it reaches threshold quicker. What do we think? Faster heartbeat, right? We're going to be able to trigger faster heartbeat. So if we think about, all right, for a certain amount of time, how many times are we stimulating the heart to contract, right? If we have a steeper, right, membrane potential that is going towards threshold, then we're going to get more stimulations of the heart to contract in a certain amount of time, right? Okay? So that results in elevated heart rate. So here we would say agents that do this would be positive or negative chronotropic agents. Positive, right? So how could we, or how does the body maybe control the heart rate in, say, decreasing the heart rate, a negative chronotropic agent? What would we do to this steepness of this curve? Yeah, so we would decrease the steepness of the curve, which means that for a period of time, certain segment of time, we would have fewer stimulations of the heart rate, or the heart to beat, right? Okay? So agents that do that are going to be negative chronotropic agents. So basically, chronotropic agents, they're adjusting how quickly that membrane potential gets to threshold, right? Everybody following me on that? Yeah. Right? So you hear positive chronotropic agents, negative chronotropic agents. Hopefully this is making sense from what we just talked about, right? And so here, a lot of chronotropic agents, the ones we really talked about a lot are the nervous system. You can start mucking with that also by changing electrolyte levels, right? So if we have less sodium outside of the cell, for instance, right? That means less sodium is able to go into the cell when those funny channels are open, right? And that would make a negative chronotropic effect, okay? Right? Endocrine system also has some modulation. We're going to focus mostly on the nervous system. And these are the two terms I told you guys about. Persisting resting heart rate above 100 beats per minute is generally tachycardia. Persistent resting heart rate below 60 is bradycardia. Right, so some terms there. All right, everybody comfortable with, the fact that we don't have a resting membrane potential here for the SA node and kind of how it functions? It's very similar to the nervous system, some of the features that we have to get the threshold and we get an action potential, and that's going to cause, instead of skeletal muscle, cardiac muscle to contract. Because we're going to talk about some of the molecular details of how this functions. All right, so sympathetic, positive, right? The major receptor, right, for that is the beta-1 adrenergic receptor. So here, adrenergic, adrenaline, epinephrine, norepinephrine, right? So what hormone or what neurotransmitter does it bind? Epinephrine, norepinephrine, right? Not surprisingly, it's a G protein coupled receptor, right? And so here, it has a second messenger. That second messenger is cyclic AMP. What was the first messenger? What was the neurotransmitter that bound to it? It is a GPCR, it's a receptor, but what bound to the receptor in the first place? So norepinephrine, epinephrine. That's the first messenger. And we have a second messenger inside of the cell. Okay? That's cyclic AMP. Turns out there's channels. Remember we talked about gating of channels? They control whether they open or close. We talked about ligand-gated ion channels, right? So the acetylcholine receptor at the neuromuscular junction, right? We also talked about voltage-gated ion channels, right? Turns out there's also cyclic AMP gated ion channels. So they open when cyclic AMP binds to them. There's other things that can also influence the opening of these channels. So here, with the sympathetic nervous system, releases a neurotransmitter, epinephrine, norepinephrine, binds to this G protein coupled receptor. Okay? causes cyclic AMP to be produced within the cell. Cyclic AMP causes sodium ion channels to open. So if we open more sodium ion channels, what's that doing to the steepness of that depolarization curve? Is it making it steeper or shallower? Steeper, right? Which means that we're reaching threshold quicker, and for a set amount of time, we're having what? Fewer or more heartbeats? More. So here we would say that's positive chronotropic, right? So this is the reason why at the molecular level this happens. Does that make sense to folks? Everybody following me? All right. Now, before we talk about parasympathetic, how many folks have heard of beta blockers? All right. Beta blockers are antagonists of this channel, of this receptor, right? So if you block this GPCR, okay, is the sympathetic nervous system going to have as much of a positive chronotropic effect? No. So individuals that have, say, elevated heart rate or high blood pressure, right, this is going to help alleviate that, right? Now, one of the problems, though, is that there's a sensing mechanism to figure out how much stimulation is coming from these receptors. So what the cells do is they figure, since they're not getting as much stimulation, they need to put more receptors on the surface of their cell. Okay? So let's say we start off with 100 beta-1 receptors on a cell, and let's say the sympathetic nervous system stimulates 10% of them, which means 10 out of the 100 are stimulated. Now we throw some blockers on there, beta blockers, and now only, let's say 5 out of the 100 are stimulated. Okay? The cell responds by trying to make more receptors available for the sympathetic nervous system to bind, so it puts more of the receptors on the surface of the cell. So we say that beta receptor gets upregulated. Okay? So rather than saying having 100, maybe now we have 200 beta receptors on the cell. Okay? And let's say that the beta blocker is blocking, some of those still, right? And the sympathetic nervous system now can stimulate 10 out of the 200, okay? Normally, without the beta blocker, it's stimulated 10%, right? So with the beta blocker, it's only stimulating 10 out of the 200 that are there. Now let's say we take the beta blocker away. Now, without the beta blocker, it stimulates 10%. So now, how many receptors are we stimulating? What's 10% of 200? 20. And originally, it was only stimulating 10 receptors, right? So we've doubled the number of receptors that get stimulated when the sympathetic nervous system is activated, right? Okay? This is very dangerous for people on beta blockers, because this upregulation means that there's more potential receptors to be stimulated if they quickly take the beta blocker away, which means that they could have a huge stimulation of what organ? The heart, right? And specifically, what is it doing to the heart rate? It's going up a lot, right? So here, the kind of the public service part of this messaging is that on beta blockers, you need to taper off beta blockers so that those upregulated number of receptors, they can be downregulated. So if you go cold turkey off of beta blockers and you're out hiking in the woods and you see a bear and you stimulate the sympathetic nervous system, that can cause a heart attack, right? So the public service part of this announcement is taper off of beta blockers. It's very dangerous if you don't. And because of this upregulation, this compensatory upregulation of the beta receptors. Everybody follow me on that? Okay? All right. So let's talk about the parasympathetic. This is coming from the vagus nerve. And remember, the sympathetic is basically using cyclic AMP gated channels to cause that steepness to increase so that we get increased heart rate? Well, the parasympathetic does two things to try to minimize increasing heart rate and also keeping the heart rate somewhat low. So here, one of the things it does is that there's a acetylcholine receptor, so it's a cholinergic receptor. One of the things that it's also is a GPCR, not surprisingly, all right? And one of the things it does is that opens potassium ion channels, right? So if we open potassium ion channels, what's that doing to the membrane potential? Is it taking it closer or further away from threshold? Further away. It's also going to be making it more challenging, or it's going to take longer time, right, to reach threshold, which means that, if we think about it, what's that doing to heart rate? Going down. So this is one of the primary mechanisms by which the parasympathetic nervous system kind of keeps the heart rate low, right? So this is the kind of vagal tone, right? The other thing stimulating the same receptor, this cholinergic receptor, it actually inhibits the sympathetic ability to open those cyclic AMP-gated sodium ion channels. And it does so, right, by basically inhibiting adenylate cyclase, which makes cyclic AMP. Right? So here, remember, in order to get that second messenger, that cyclic AMP, that G protein activates adenylate cyclase. Right? So I didn't draw it out over here. But in order to get cyclic AMP is made by adenylate cyclase. What the parasympathetic is doing is that it inhibits adenylate cyclase, which means that we're not making as much cyclic AMP, which means that we're not doing as much of opening those sodium ion channels. Everybody following me on this? So the parasympathetic does two things. Causes potassium ion channels to open, and it's interfering with the ability of the sympathetic nervous system to make as much of that second messenger, right? And that second messenger is important for opening the sodium ion channels. Okay? So there's two things the parasympathetic does to interfere with the ability to increase heart rate. So I see some puzzled looks out there. Yeah. Only when you're being, out in the chased by someone. Yep. Because remember, when we have dual innervation of organs, we have something called tonicity. What has a dominant tone? We say during the normal state, right? Here, the dominant tone is parasympathetic. So there's tonic firing of the vagus nerve. So here, during, normal state, we have mostly this going on, right? If you're out being chased by a bear, then this gets activated, and it overcomes this. You following me on that? All right. So in order for it to elevate heart rate, one of the things that happens, there's actually two things that happens. One is we stop the parasympathetic, right? So remember the parasympathetic, there's vagal tone that suppresses heart rate. So even we can increase heart rate just by decreasing parasympathetic, right? Because it's putting the brakes on the heart rate. The second thing is that we start stimulating the sympathetic, which means that we're elevating actively the heart rate by opening those cyclic AMP-gated sodium ion channels. There's two things that can go on to help elevate the heart rate. Everybody following me on that? Okay. Right? So this is the molecular events that are going on. It's kind of this competition for the membrane potential, right? Is it going to be quickly reaching threshold, or is it going to take longer to do so? More quickly it reaches threshold, the higher the heart rate. The longer it takes to reach threshold, the slower the heart rate. So positive and negative chronotropic agents. How are people feeling about that? Is that making sense to folks? Any questions? People understand this competition of cyclic AMP between sympathetic, parasympathetic? So the cyclic AMP opens sodium ion channels, right? So sympathetic is going to promote the formation of cyclic AMP, which is promoting the opening of sodium ion channels. Parasympathetic is inhibiting the formation of cyclic AMP, which is inhibiting the opening of sodium ion channels. All right? Feeling better? Yeah? Okay. Okay. So chronotropic agents, there's a few out there that you can add to your body, right? So nicotine and metabolites thereof. So coteine's a major metabolite of nicotine, and this stimulates catecholamine secretion. So catecholamines, right? So those are epinephrine, norepinephrine. So is that going to be a positive or a negative chronotropic? Positive, absolutely, right? Positive chronotropic agent. Thyroid hormone upregulates adrenergic receptors. So adrenergic. So what is that? A neurotransmitter or a hormone? What are you talking about? Epinephrine, norepinephrine, adrenaline, right? Okay. So is that going to be positive or negative? Positive chronotropic. Caffeine and related compounds. So it turns out they inhibit cyclic AMP breakdown. So there's a molecule called phosphodiesterase PDEs. They break down cyclic AMP, right? So remember, if we activate a system, we have to have a way to reset it to baseline. So if we make a bunch of second messengers, we have to get rid of those second messengers when we don't want them. So there's an enzyme that does that. Turns out caffeine and related compounds inhibit the breakdown of cyclic AMP, which means what's happening to cyclic AMP levels? Are they going up or down? Yeah, so if we inhibit the breakdown of them, then there's going to be more of them, right? So if we increase the amount of cyclic AMP, right? So it's kind of like the sympathetic nervous system, right? So what's that doing to the heart rate? It's going up. So is that positive or negative chronotropic? Positive. So before we get off of this PDE thing here, turns out there was a clinical trial being done for a PDE inhibitor. It's supposed to treat, individuals with cardiovascular-related issues. But a lot of the men in the trial kept coming back for it, right? And one of the reasons was it actually was inhibiting a PDE that is found in the penis. And so we know that as Viagra. So, I'm sure a lot of the clinicians and the people in the study were like, why are these men coming back? So, anyway. So there's different PDEs in different parts of the body, and so if you have knowledge about the different structures of the PDEs, you can design pharmaceuticals to target them. All right? Okay. Other things that can affect this, and this is really critical for those that are monitoring, say, patient vitals. Right? So here, potassium ions, right, are really important. So potassium ions have a huge effect on the heart rate, huge chronotropic impact. Think back to what we talk about. In order to repolarize, we open potassium ion channels, right? And also for the parasympathetic, one of the things it does is that it opens potassium ion channels in order to get the vagal tone, the parasympathetic tone on the heart. So potassium ions are really important with respect to chronotropic agents. Calcium, we saw, also is important, right? And can affect heart rate. But as we'll see, it also affects contractility. So the contraction of the cardiac muscle tissue, it actually is kind of similar to what we saw with skeletal muscle. How important was calcium with skeletal muscle contraction? Yeah, it was critical, right? If we didn't have calcium, then we didn't get the cross-bridging to go on. Right? So calcium has a huge impact also on contraction strength, what we say contractility, right? So the term that I threw out earlier, agents that affect contraction strength, are called inotropic, I-N-O, tropic, okay? And so here, calcium's more of an inotropic agent than it is a chronotropic agent. So you can impress your friends with your newfound terms. Okay? Inotropic. Right? So here we've now seen calcium's really important for heart contraction rate. Also has an effect on chronotropic. Also is important in the clotting cascade. Right? Also is important in bone metabolism. Right? Bone structure. Right? So all of those things. So here we're starting to see this overlap of these different substances have wide-ranging impacts. They interconnect with each other. Okay. So last slide and we'll take a break. Some differences between the skeletal muscle and cardiac muscle. Right? So here, this autorhythmicity, so ability to spontaneously, rhythmically fire. So we're talking about the SA and AV nodes here. They don't need nervous stimulation, nervous tissue stimulation, but the nervous system can impact the rate at which they fire, right? Parasympathetic versus sympathetic. These autorhythmic cells, they can depolarize the entire heart. And so here, unlike skeletal muscle, cardiac muscle tissue's connected. So here, the tissue, the cytoplasms of neighboring cells are connected, so if one gets the signal to contract, its neighbors are also getting that same signal. So they contract kind of as a unit. Right? All right. So all contract as a unit or none do. Long absolute refractory period. One of the major differences between skeletal muscle and cardiac muscle. So remember here with skeletal muscle, we had a really sharp, membrane potential, okay, for action potential, right? With cardiac muscle, it's really long. One of the reasons for that is so that when we have contraction, we're not going to get another contraction for a little bit. So it makes sure that we don't get, say, abnormal contractions, right, that happen haphazardly. So here, the other part is the tension that it generates, right? So here, the tension we get from skeletal muscle, right, is really quick, but it's sustained with cardiac muscle. Why might that be important? Why would we want to sustain a contraction? Think about what the purpose is of the heart. Yeah, it's going to be more efficient pump to do that, right? So if you just did a quick pump, you may not eject as efficiently. Right? The other thing that helps is to maintain that directionality. Because remember, it's not a good thing to have blood flowing back from where it came from. We want to keep it flowing in the same direction. All right? That makes sense, the differences between skeletal and cardiac muscle. You're going to want to take a look at this. All right? So we've talked about chronotropic agents. We've talked about some of the differences in skeletal and cardiac muscle. Is that making sense to folks before we go for a break? People feel good? All right. Okay, so why don't we take a break? And after the break, we'll pick up with looking at a little bit more of ECG, and it'll help you guys in the lab. All right, so I think we'll continue our discussion here. And what I'm going to want to make sure to get through is talk a little bit about ECG, give you guys a bit of an intro into talking about blood vessels and peripheral resistance a bit. And then I want to talk about blood pressure. And so that means we'll skip over capillary exchange, and we'll revisit that on Monday, because I think talking about blood pressure and cardiac output, peripheral resistance, will help you guys with the discussion homework. All right, so that's kind of what how I want to reshuffle things. You're going to get a lot more about the details of the ECG trace from Dr. Jack McKenzie in lab. So I'm just going to go over some basics here of it for right now. Now remember here, we're talking about the cardiac conduction system. Remember, that's different than the cardiac cycle. Cardiac cycle is a physical movement of blood through the heart. Here, the conduction system is the electrical impulses that are going through the heart that will lead to the physical movement of blood through the heart. All right? And so here, the ECG, what it does is it picks up on those electrical signals. All right? So we start off, right? The SA node starts firing. We get depolarization. And we actually get just a flat line at that point because only a small amount of that tissue is being electrically stimulated. Not enough is being stimulated for that electrode that far away to pick it up. All right? Now, as this depolarization event spreads, more and more of the tissue is being depolarized. So eventually that signal is going to be able to be picked up by the electrode. Right? And so here, that's what we're starting to see with this deflection on the ECG trace. So what part of the heart is starting to depolarize? What chambers? Atria, right? So it's starting off in the right atrium, like you guys said, and then it's spreading, right? The more tissue that the depolarization happens to, the larger the signal, the greater the deflection on the ECG trace. And so here, once we get all the atrial tissue to be depolarized, okay, then it's going to go back to the baseline. So here, this part of the wave is called the P wave. Okay? And so P wave is indicating what part of the electrical conduction system. So what do we see in depolarizing? What part of the heart? Atria, right? Okay. The other term I want to introduce to you is some orientation of the heart. So up here, we call this the base, and then where it comes to a point, we call that the apex. So here, this whole ECG trace is tracking the depolarization event that goes roughly from the base to the apex. Okay? But we start off, that P wave is depolarization of the atria. Right? Now, where's that electrical signal going to go to next? Where's its next stop? Where's its next destination here? AV node, right? So it goes to the AV node. Okay? And it pauses a little bit there, so that's part of what's going on here. Okay? And then that signal is being sent down via the bundle of His and then these interventricular branches. Remember that there's that bundle and then it branches so that you have a branch that's going down the interventricular septum lining the right ventricle and the part of the interventricular septum lining the left ventricle. Okay? All right? And so with this depolarization event that starts going on, we start seeing another deflection in that ECG trace that's picking up the electrical signal. Right? And so this one was the P wave. This one is the Q. Right? And now, what part of the heart are we starting to depolarize here? What's that? The ventricles, right? And specifically, not all of the ventricles. We're starting kind of inside, and then we'll kind of work it out a bit, right? So here we're starting to depolarize in here, and we're starting to pick up because a lot of that tissue is being depolarized enough such that we can pick it up on the instrument. Okay? So here we're starting with the depolarization of the ventricles, starting there in the interventricular septum. All right? So we start that's the Q wave. Now, as the electrical impulse spreads, it spreads through a whole bunch more tissue. The more tissue that gets depolarized, the larger the electrical signal. So not surprisingly, how does that represent on the ECG trace? Well, if we have a little deflection with a little bit of tissue that gets depolarized, if we have a lot of tissue, what type of impact on the deflection do you think that has? It makes it bigger, right? So not surprisingly, we get this huge event, right, when we have a lot of that ventricular tissue depolarizing. So here we had the P wave, then we had the Q wave, and this is the R, and then the S. Okay? So here, the Q, the R, and the S are all part of ventricular depolarization. Okay? So we sometimes refer to this as a QRS complex. What is it representing? So what? Cardiac conduction event. Ventricular depolarization. Now, ventricular depolarization, what cardiac cycle event is that going to lead to? Systole of what? Ventricles, absolutely, right? So this is what you want to do. You want to start connecting what's happening on the ECG trace, the cardiac conduction system, and the mechanical events of the cardiac cycle, right? Which one happens first? Yeah, so basically the cardiac conduction system, right? That's leading to the physical, contraction and mechanical movement of blood through the heart. That precedes the actual ventricular contraction. So here, if we're taking a look at what's happening over time, we see the electrical signal for ventricular systole, right? Or depolarization, excuse me. Ventricular systole, though, is happening a little bit after that, right? What about here with the P wave? We got atrial, what? Depolarization, right? Where is the atrial systole happening? Slightly after that, right? So remember, the actual physical contraction happens slightly after the electrical stimulus happens to promote that. Just like when we talked about skeletal muscle. We don't get skeletal muscle contraction happening. We only get that happening after the electrical signal from the motor neuron. No, we don't have motor neurons here, I understand, but the same type of principle. Electrical signal precedes the physical contraction. Everybody follow me on that? All right. So during this time, when we get ventricular systole, okay, ventricular depolarization, the atria repolarize. Okay? Now, we don't pick that up because the ventricular depolarization is such a huge event. So remember, this ECG trace, as Dr. Jack will tell you, is a summation, right? So it's a summary of what the electrical activity is happening in the heart. Electrical activity that's coming from the ventricles is so much larger than what's happening at the atria at the same time. So you don't see it. It's kind of masked by this huge depolarization of the ventricles. All right? Now, we've done atrial depolarization, and then we did repolarization, then we did ventricular depolarization. So what happens next? Ventricular repolarization, right? And so that's the next event that happens. All right, oops. And I don't have that on here. Okay? All right, so there's a T wave right here that is going to be the ventricular repolarization event. Okay? All right, so there's going to be a P and then a QRS wave, and then there's a T that's going to be a repolarization of the ventricles. Okay? And then for another round, we'll see the same trace again. Everybody following me on this? The ECG is picking up the electrical signals from different parts of the heart. And you can tell what's happening in different different electrical conduction system, what's happening in the conduction system of the heart by looking at the ECG trace. And what's happening in the electrical conduction system, the cardiac conduction system here, is leading to what's happening in the cardiac cycle. Everybody follow me on that? Okay. You'll get more of this in the lab. All right? Okay, blood vessels and circulation. So we're going to talk a little bit I'll give you an introduction a little bit here, and then, like I said, we'll skip over the capillary exchange and revisit that on Monday, because I want to talk a little bit about how to, what influences blood pressure. Now, here, some definitions. Arteries carry blood away from the heart, veins towards the heart. All right, capillaries are where we get exchange of material such as gases and nutrients, and then we're going to have slightly smaller arteries and veins. Slightly smaller arteries are called arterioles, slightly smaller veins are venules. Arterioles are also known as peripheral resistance vessels because those are the ones that largely can contract and change their diameter, and that's going to influence blood pressure greatly. All right, so just keep that in mind a bit. Okay. Now notice here that we have blood going out from the right ventricle towards the lungs. So if it's going away from the heart, right, is that a vein or an artery? Artery. Now that blood that's going towards the lungs is less oxygenated. So here, less oxygenated blood in an artery, especially when we talk about the pulmonary circuit here, okay, right? We don't necessarily talk about arteries always having the more oxygenated blood, right? It's a common misconception. Now, blood that's been been oxygenated and then is returning back to the heart, right? So what vessel is this taking blood back to the heart from the lungs? A vein. And here, this is more oxygenated blood, but it's a vein. So here, this idea of having more or less oxygenated blood in veins and arteries doesn't hold. We only are classifying veins and arteries then dependent upon whether they're taking blood to the heart or away from the heart. So keep that in mind. Everybody make sense, folks? All right. Okay, so here, blood vessels, similar to what we saw with tissue, in 231, we have kind of a epithelium that's made up of cells that are called endothelial cells. So this is also called the endothelium. And remember, if we have an epithelium, underneath that, attaching that epithelium to underlying connective tissue, we have the basement membrane. So remember, we have epithelium, basement membrane, connective tissue. And this opening, right, is called the lumen, right? Okay, so lumen. So you can see here in this upper picture, right, we have a lumen of an artery and a lumen of a vein. So take a look at the blood vessel wall. Which one's thicker? Artery, absolutely. So why do you think it's thicker? Blood pressure, absolutely. It's higher pressure system. The arterial system's a higher pressure system, as we'll see. Venous system's not so high. Okay, so it doesn't need to have as thick of a tissue. Now, tissue making up its blood vessel wall. Now, one of the other things to point out here is that we have smooth muscle. Right? Not as much in the venous system, a lot more in, say, the arterial system. Which means that when smooth muscle, what can it do? It can contract and relax. So here we can control the diameter of blood vessels, much more in the arterial system than in the venous system. This is going to be important in regulating blood pressure. All right? The other thing is some of the larger arteries, like the aorta, they have elastic tissue in them, because they get a huge surge of blood pressure that's coming from the heart, because the aorta is really close to the heart. So it's got to be able to go and contract back to its normal resting size, right? So it has some elastic tissue there as well in some of these large arteries that are close to the heart. Okay? All right. Okay, so this is just telling you a little bit. So elastic arteries, like I said, have some elastic tissue. We have some muscular arteries that have more smooth muscle, and arterioles that have relatively a high percentage of their vessel wall is smooth muscle. Okay? And we can control a lot of resistance here. And then we have the smallest ones are capillaries, and there's different types of capillaries. We won't go into the details. Some allow more exchange between the blood vessel compartment and the surrounding tissue. All right? And so we'll circle back to that exchange, like I said, on Monday. And then with the venous system, not surprisingly, you don't see as thick of walls there, and you don't see as much smooth muscle in that area. Okay? Now, some terms here. There's 3 general layers for blood vessels. They're called tunicas. The one that's making up the area that's closest to the lumen is called the tunica intima, and then surrounding that is the tunica media that has mainly the smooth muscle, and then we generally have some supportive connective tissue around that that's called the tunica externa. Right? So we kind of group some of the different regions that make up the blood vessel wall as the tunica intima, tunica media, and tunica externa. All right. Tunica media has most of the smooth muscle. All right. Okay. So this is pretty much what I was just telling you guys about. We have the externa, the media, and the intima, and then the lumen, that's the central space where we have the blood fluid and blood connective tissue. All right. Venous return. So here, because it's a low-pressure system, right, there's not a lot of pressure to help with moving the blood through the venous system, okay? But there are certain things that help. So the muscular pump's the main one. Skeletal muscle that are on both sides of the veins, all right, can help milk blood through the venous system back to the heart. So, pilots, fighter pilots, jet pilots, right? That have a lot of G's pulling their blood towards the bottom of their body. One of the things they tell them is to start flexing their skeletal muscle on their legs. Why? Because it helps with trying to return the heart or blood back to the heart. Okay. There's also respiratory pressure changes during breathing. So when your cavity expands and then contracts, it also kind of has this milking action that kind of pulls blood in that direction. Venoconstriction, so there's some, sympathetic innervation that contracts the venous system, not as much as with the arterial system we'll see. Certainly gravity, so here we're not talking about gravity helping blood return to the heart from the lower extremities, but certainly from places above the heart, if we're standing up, for instance. And then venous pressure gradient, so it turns out that venules have higher pressure than, say, that the venous blood vessel closest to the heart. So substances move down their pressure gradient, just like moving from high concentration to low concentration. So venous pressure gradient helps. And then the pump action, constantly moving blood out of the heart, kind of has a cardiac suction effect. Okay? So all these kind of help. The skeletal muscle does a really good job, though, that milking action. Does that make sense to folks? All right? Okay. All right, so this is just a bit of a summary between arterioles and veins that we just talked about, so be sure to take a look at that. And like I said, I want to skip this and talk a little bit about this whole idea of blood pressure. We'll circle back to the capillary exchange on Monday. Blood pressure. It's a combination of cardiac output, how much blood's coming out of the heart, and then how much resistance is found in the blood vessels. And in particular, we're interested in the output from which side of the heart. Which one's going to systemically pushing blood, the right or the left? The left. So we're really kind of interested in the left cardiac output, and then also systemic or arterial resistance, peripheral resistance, right? And so here, peripheral resistance and cardiac output are the two major factors, okay, that influence blood pressure. All right? So here, cardiac output and peripheral resistance. So let's talk a little bit about peripheral resistance. So it turns out you can change resistance by changing the diameter of the blood vessel. Specifically in the arterial system, that's the arterioles. Okay? And it is exponentially related. So here, if we increase the distance, right, of the tube that something has to flow through, okay, right, that increases the resistance, right? So it makes sense. The longer the tube, the more resistance you encounter. You can also change the resistance by changing the diameter, right? And this is actually, if you do inversely proportional for almost 1/4, right? So here we're saying that if we do change the diameter a little bit, we have a huge change on the resistance. So it's not a linear effect. So changing the diameter has a huge impact on resistance. Okay? And so here, longer the vessel, greater the resistance. The diameter has a massive effect. And that's one of the major ways we can change peripheral resistance, is changing the diameter of arterioles. Certainly, thickness of blood can also impact blood pressure, right? So here, if we have, say, a huge amount of EPO, stimulating a lot of production of erythrocytes, that makes the blood thicker. It's increased resistance. Okay. Now, the other one is cardiac output. Now, if you remember, I told you about stroke volume. There was milliliters that was ejected per each contraction, right? And if we factor in heart rate, right, if we say, okay, the heart beats this many times per minute, and for every beat we're ejecting this amount of blood, we can multiply those two things together to figure out how much blood gets ejected per minute, right? That's the cardiac output, okay? That's the cardiac output, the liters per minute. All right, so here, remember heart rate times stroke volume gives us the cardiac output. Now, let me write a few things on the board that hopefully will help people out. All right. So blood pressure is cardiac output, and then we'll say that's times peripheral resistance. Okay? All right. Cardiac output right? What was that? That was heart rate times stroke volume, right? That was the cardiac output, right? Okay. And if you look back to stroke volume, how did we figure that out? Well, that was end diastolic volume minus end systolic volume, right? Makes sense? Remember, this was how much blood was in that ventricle, right, before it was ejected. And this is how much blood was left in the ventricle after systole, right? That was our stroke volume. So here, what you want to be able to do is say, okay, well, what if we, say, increase end diastolic volume, right? Okay, what's that going to do to stroke volume? It's going to go up, right? And if everything else is the same, what's that going to do to cardiac output? It's going to go up, which means what's happening to blood pressure? It's going to go up. This is what you want to be able to do. All right? Let's do one more, and I'll let you guys go. All right? Okay. Okay, so let's say we decrease, right, end systolic volume, right, and we're going to increase heart rate. Okay, so let's start here. End systolic volume, we decrease it, which means what's happening to stroke volume? So if you're ever confused, just throw some numbers in there, okay? So here, let's say we start off with, I don't know, 8 and minus 4. Okay, that was the initial situation. Then we decreased end systolic volume. We didn't do anything to anything else. So let's say we have 8, and let's put a number less than 4. 2, right? So which ones would we do? Increased it, right? Okay, so we increased stroke volume. So what does that happen to the cardiac output? Increase, which means increased blood pressure. All right? Make sense to folks? All right, I wanted to make sure to show you guys that for the homework. Okay? All right. Thank you guys for sticking with me on that. And then, like I said, we'll circle back around to capillary exchange on Monday. All right? Okay, I'll see you guys up in the lab. We got a lot going on in lab, so try not to be late. Okay, I'm going to open the doors at 10.